So thank you very much. It's really great to be here and also really special to talk to, a group of population genetics, quantitative genetics. Like typically the audience is a little more mixed. It's particularly delightful for me to be here in Vienna. Um, okay, I like to always start with my acknowledgements just because I don't like to run out of time at the end. And just to say that I I don't like to work by myself, and everything I'm doing is in collaboration with many other people, and that I really appreciate the opportunity to collaborate with people, and that this work that I'm going to talk to you about today is, as Christian alluded to, kind of an interest of many years of mine, and with a lot of different people. And I will be highlighting different individuals throughout the talk. Um, just to give some background to the things, the questions that I'm interested in, is I like to um, basically talk about kinds of systems genetics, or Richard Janssen used to call it genetical genomics, but basically the concept that you are thinking about phenotype is genotype plus environment and the interaction, but a lot of the phenotypes that we're thinking about are in fact gene expression, how to connect expression to proteins, expression to organismal phenotypes, DNA, alleles to expression, and so on. And in particular, I have been interested in allele specific expression and also splicing, which I don't talk about today. Um, so the really fun part of what I will talk about today is kind of why do we say genetical genomics is typically because we're looking at some kind of crossing schemes where we're making deliberate combinations of individuals, um, and we do it in different ways, and this gives us different kind of genetic structures that we can play off of. So so I think this has occupied me for many years, um, and hopefully will be interesting to you guys. Um, I guess the first thing to say is maybe a little bit obvious, but transcription is complicated. There's a lot of things going on, um, and we can loosely partition this into two things. We might call them cis effects and trans effects, where the cis uh, somehow I did something I skipped. Okay. We have cis effects and trans effects, where cis effects are really the things in the red box, the promoters and enhancers, the things kind of attached to the gene itself. And then the trans effects would be like all those other proteins that come together in the complex. We can think about this in this way. This is complicated, but there's no genetic variation in this picture at all. And there's also only one allele in this picture of two or more. So this is problematic because as complicated as this is not even close to kind of any approximation of reality as we might think about it. Um, okay, so if we want to talk about cis variation, we really want to define this as genetic variation in the regulatory region of the gene. And then trans variation is in those little regulators of expression. Oh, and now we're automatically advancing. Great. Okay, so if there's no cis variation, then you will not see any trans effects. So just think about that. I want everybody to sit with this just for 1 minute. Like, if you do not have cis variants, you can have all of the trans variation that you want, but you won't see it because you need to have the cis variation in order to be able to measure the difference. So how are we measuring this difference? We're going to use allele imbalance. That is, we're going to look at the difference in the frequency of the transcripts of the orange versus the blue alleles. And we have some hypothesis that they should be 50-50. And if they're not 50-50, then we're going to say, okay, there's evidence that there is a difference. So Allele imbalance is due to genetic variation in cis regulatory regions. And we can see this at the locus itself, but we can also see this when we have interactions between trans factors and the cis regions. So why is this important? It's basically important because we have lots of complicated human diseases that we know have to do with regulatory variation, which is the beginning thing that I used to say to all of my grants, that one used to get funded, but now we'll see. Okay, small joke about the current situation in the United States. But it's also a direct measure of the impact of genetic variation on the phenotype. Okay, in this case, the phenotype is gene expression, and it's really widespread, not just among diploids, but among polyploids. And this can be really interesting when you start to think about this in terms of the polyploids. Okay, so you have these kinds of difference with copy number variants that can happen in diploids and in polyploids. So in order to do this, you need to be able to measure your alleles separately. That means you need to have polymorphisms in the transcript. You need to be able to see the SNPs in the sequence that you're measuring. That's not the same thing as the causal variants, which I think everybody in this room probably understands better than most. Okay, but you need to have these, let's say, tracking variants. There are some complications when you want to talk about allele imbalance. Okay, first of all, there's something that people loosely refer to as map bias. And that's actually a big tent word. And one of the things, I don't know why it's one of the big things is to think about the choice of the reference. So you can use a common reference, or you can have personalized references. You can also think about the quality of the reference. You can also think about the mapping algorithm that you use. And you can also think about actually the sequencing itself potentially having biases. So all of those things kind of together, people refer to these collectively as map bias. Something that I think is important to just state is your intuition may be that, like, if you had some kind of small bias, so 5%, that would mean that instead of an estimate of 50%, you have 0.525. It feels kind of small. That actually can result in a type 1 error of like 50% when you have just sort of moderate levels of gene expression. So really small biases can really get amplified in allele imbalance studies very, very quickly. And this has really been demonstrated time and time again. One of the things that really contributes a lot is mapping to a common reference as opposed to a specific genotype reference and calling your SNPs off of the common reference. And here I'm just going to show you, and it's not the details are not so important. It's just to say, like, at the 0 line, where that red line is on the left panel, which is DNA, on the right panel, which is RNA, that if you map to a genotype specific reference, your DNA, which you would measure as the frequency 50/50, will be centered, although not completely unbiased, will be centered around that 0 line. Whereas when you map to a common reference and use the SNPs, you are not even centered on the 0 line. So this is a big problem, particularly when you're going to have this type 1 error that gets really big really fast. And it's mirrored in the DNA and the RNA. So it's because when you sequence the cDNA, so it's going to be the same. Okay, now I am going to make a plug to Christian, special for you, for long reads. Um, so my group, we started six, maybe 6 or 7 years ago to really think hard about long reads and to think about how long reads could really help. And they really do help a lot. So you can think about two ways of mapping if you're interested in doing allele-specific expression. In one way, you map basically to haplotype 1, and then you map to haplotype 2, and then you compare your mapping scores and you say, okay, do I have one is better? Or you can map to the genotype-specific reference. And either way, we've actually demonstrated with Nadia Nolte, we've demonstrated that you get the exact same answer as long as you count the reads that map non-specifically, and then you count the reads that map specifically to the haplotypes. That's fine. Why long reads? Because you're going to get many, many more reads that map haplotypes specifically with long reads than with short ones. And you are going to get the phase, okay? So they make a huge difference. You get more of them and you have more phase. So you have fewer reads that are unassigned than with short reads. With short reads, you're going to have a lot of reads that end up unassigned. Okay, so long reads. You have a lot of these problems, and I'm just going to kind of tick through some of them. For example, you're missing SNPs in your reference. So even though you've done a personalized genome and you've got all your haplotypes, you don't get all the variants. You miss SNPs, you miss structural variants, you have problems where you miss them. You can also, when you start to think about haplotypes, I alluded to polyploids, but even in diploids, you can have copy number variation. You can have different loci that have different numbers of copies. And you can have different copies between haplotypes. These make things complicated, and you can miss that. You can also have length differences in haplotypes due to misassembly and misannotation. So when you're looking at this, the length of the genes looks like it can be different. And then you can really think about these reads that are multi-mapping. And if you follow a default pipeline, a lot of these with map quality 0, like they will just disappear. This is a problem because those multi-mapping reads will be an estimate of the expression, but also can help you estimate like where you may have missing copy number variation and things like this. Okay, so just very quickly, 3 kind of examples of how long reads can help you, and something we've spent a lot of time on. On the top, we're looking at, I'm just going to say that the very top left, and the last time I tried to push the laser pointer, I sent myself forward. So just look at the top left. The types of bias you can have would be missing SNPs. So next to the fruit fly. Okay, good. Thank you. Perfect. Okay, so on the top left, here. Okay, next to the fruit. Next to the fruit fly, we're going to talk about missing SNPs. So if you're missing SNPs, what you may see is here's the distribution in the middle here. Instead of being centered around the 50%, you see it looks kind of funky. Very official description, but it's not symmetric. And this lets you know, like overall, systematically, you're missing SNPs. You can actually use the long reads to find the missing SNPs and then to correct your underlying reference. And then when you do, you can end up with something that is more centered. Okay? So this is even without the DNA. You can just do this on the RNA. You can find your missing SNPs and you can fix. Okay, here, what if you're missing copies of the gene? You have multiple copies of the gene and you don't know you had it and you're missing them. You can see this on the edge by having things that look like there are very extreme allele bias, like things that are like close to 100%, 85% bias one way or another. Is a way of actually saying I have too many copies of this and I didn't know it. So if you go back and you look for those copies, the long reads will let you actually distinguish between copies of genes as long as they're not immediate tandem duplicates that are identical. If you have any divergence between members of gene families, you can find them and deconvolve it. So when you do that, you can get rid of those little extra points. And another thing that you can do here is if you have this issue with length differences, again, you're going to kind of see this spiky distribution that you can reduce by identifying which peaks are outside of your expected sort of central area. Okay. So I think long reads really help you identify places where you might have this kind of umbrella map bias, by which we mean, kind of all of these things that could be going wrong. All right, so now I have my first take-home message, part 1. Pay attention to the technical details. This is really important when you start to deal with expression data. Things like personalized references, multi-mapping reads, systematic bias, looking for things that are extremely biased. We've done more than a decade of allele-specific expression studies. The vast majority of what we see is quite moderate. So when you start to see extreme bias, you want to investigate that, make sure it's not something that is an artifact. And long reads really make everything just a little bit easier, like to identify problems in the data. Okay. But testing AI actually itself, there are also some complications. So those of you working with expression data probably have been hearing about overdispersion. I want to tell you that part of this is that reads are not actually predetermined. Like you don't specify for this gene, I would like 5,252 reads. Okay, so the variance over dispersion is coming from the fact that you're sampling. And then you're also going to have a different level of precision when you have different expression levels. Okay, so you're going to have more power to detect allele-specific expression when you have higher coverages that let you, and more SNPs that let you basically separate the alleles. So this was the problem that converted me into a Bayesian approach, basically because the reads are random. There is really no way around it. You are sampling, and that sampling variation is random. When you take this, even for kind of a basic RNA-seq analysis, and if you use kind of a random sampling approach, you will see that you instantly fix all the overdispersion. And in fact, it doesn't matter. We've played with all kinds of distributions. So use like Poisson gamma, negative binomial. You'll get very, very similar inferences. So you're not really sensitive to the distribution here, but what you are sensitive is to the fact that there is a distribution. And I will also say it helps you a lot in any particular case if you have DNA, because if you have DNA, you can actually look and see, well, how is that behaving? So if you think you have DNA, it should be 50-50 and a diploid. If you're departing from that in the DNA, then you can adjust. What do I mean? So if I'm looking at my bias in my RNA and I'm like, oh, look, I have 80% towards this one allele, or I have 26%, that looks really extreme. It couldn't be a technical artifact, right? But if I look at the DNA, I have exactly the same. So what is that? Can be all kinds of things. Can be hidden structural variation, can be copy number, can be whatever. But if you have the DNA, you can actually include the DNA information as a prior, and then you can get a corrected estimate of your expression. Okay? Okay. The thing I want to talk to you about mostly today is the environment. Okay, so I'm interested in gene bioenvironment interactions. I think a lot of people in this room are interested in gene bioenvironment interactions. And so when we think about the environment, we could think about a lot of things. We can think about tissues, we can think about cell types, we can think about temperature, we can think about in Drosophila female mating status. You can think about reciprocal crosses, for example, that have the same DNA. Okay, so you can think about a lot of things as affecting the environment. I'll just talk briefly about the idea that you have the same genotypes, but you have virgin females and mated females. Why did I pick this one? Well, because if you actually think about virgin females and mated females, like the gene expression differences, and oh, I have the guillotine to remind me to tell you I'm talking about their heads. So we cut off their heads and then we look just at their heads. It's like some way of saying, oh, we have something less complicated than the whole fly. So we have their heads. Now, I would have exactly the same genotype, except I have virgin females or mated females. Somewhere between 3,000 and 5,000 genes have a difference in expression between mated and virgin females. That's an enormous difference. So now if I also want to ask, do I have differences in allele imbalance between the mated and the virgins? Why would I want to do that? Well, in one status, one environment versus the other, I'm going to have all these different trans factors, all this different trans stuff. The cis is exactly the same. So remember I said you can only find trans if you have cis, but now I create a situation where I have the same cis, so now I can look for trans. Okay, great, between the two environments. But now I have to actually test statistically whether the difference in the allele imbalance in one environment is the same as the difference in allele imbalance in the other environment. Okay, so the way we're going to do this is we're going to go back to Mr. Bayes, but we're also going to be thinking about the environment, and we're going to be thinking about the kind of non-specific gene expression. And this is important, right? Because I said, okay, if you have allele imbalance, I'm going to have more power if I have more reads. So now I'm going to do a difference in gene expression, 3,000 genes. Some of them are going to be more expressed in one condition than the other, and I'm going to confound that with my power. So I need to be able to address this. I need to be able to consider how much that gene is expressed and the difference between that gene in the two environments, and account for that before I actually do my statistical test. So we can put this into the model, and actually we can also put in the map bias, and allow that to be different between the two alleles even, and then do a test. Okay, so now it's really getting complicated, right? It's really complicated. Like you have a lot of steps. And I will just say that at some moment I was extremely fortunate that not only did I have this really wonderful undergraduate in my group, I had an entire uh, army of undergraduates who were very interested in this, and they built a pipeline and put it in Galaxy. And so all of these kind of steps have been delineated and mapped out, and all the code is there, and so it's all beautifully done thanks to them. Okay, so my take-home message now in part 2 for testing differences in allele imbalance is you're going to have overdispersion. This is because reads are random, so you really need to be thinking about a Bayesian approach. Differences in expression levels are real, and when you want to compare between environments, you're going to also need to take that into account. I'm going to just say this again, maybe 3 more times before we're done. Do not throw away your multi-mapping reads. Do not throw away your multi-mapping reads. Do not throw away your multi-mapping reads. Even if you're doing short reads, okay, you're going to create a lot of biases when you throw away your multi-mapping reads. Okay, and we have a statistical model that's going to help to tell the difference between the environments. All right. Now, let's talk again about trans factors. Let's talk more about trans factors. Notice here, I actually have, it's like a reciprocal cross. What have I drawn? The actual alleles are the same. The cis is the same. The trans factors are different, right? So in one case, I have a bunch of blue trans factors. In the other case, I have a bunch of orange trans factors. So now I'm going to have this test, again, comparing the cis is the same. So the only thing that's going to change this is going to be the trans and that trans interacting with the cis. So that's really good. I can also do it this way, okay? I can also actually compare if I have 2 different setups where I have one shared allele. So here I'm sharing the orange allele, and the blue allele is different. I'm sharing orange, blue is different. Now I can compare allele imbalance in the first case versus allele imbalance in the second case. What is that? That's the cis effect, but also the cis effect interacting with whatever trans differences there are. Why would I possibly do this? Why would I? Because remember, I was like, oh, I can compare the 2 alleles within the individual to get the cis, and then the trans is constant. So why would I do this? Anybody want to guess? There's a trans difference, yeah, to match the trans difference. That's exactly right. Because here, I'm going to have more power than here. Because this is going to be between 2 individuals, and this is within an individual. So you can already imagine that if you have this conversation, do I have more cis? Do I have more trans? Where is it coming from? This is a test within individuals, and this is a test between individuals. So what I really want to have is 2 tests between individuals. This test for cis between individuals, and this test for trans between individuals. So now I have the exact same statistic, and I can test cis or I can test trans. Okay. Now it gets really fun. All right. So remember I was saying, okay, in the first case, we are just looking at this within the individual. Now imagine that you are doing a recombinant inbred line. So you have a recombinant inbred line, and you cross that to a tester. When you cross the recombinant inbred line to a tester, you immediately basically create a balancer. So what you end up with then is that if I have two recombinant inbred lines that share the same cis region and I cross them to a common tester, then when they share this, this is going to be, and I compare the allele imbalance between these two, this is going to be a test of the trans factors. Whereas if I have the recombinant inbred lines and they have different, two different lines have different alleles, then when I compare them, it's cis. So that means for the same cross, when I compare a set of crosses, so I take recombinant inbred lines and I cross them to a tester, I can look at the same pairs and compare those pairs. So I can and ask the question whether it's cis or trans, just depending on whether the haplotypes are shared or different at the particular locus that I'm looking at. So this particular one is an 8-way or 16-way, really. I think that's a good question. And one of the nice things about the long reads is it lets you actually see your haplotypes on the as you're tracking. But it's true. I mean, but I think you can also decide like if the haplotype in a region is consistent. In other, you know. Yeah, I mean, this particular set of recombinant inbred lines, I think the blocks are pretty big. So this is the DSPR from Tony Long and Elizabeth King. So the blocks are fairly big. I think it's 20 generations. I have to go back and look. But so I think the blocks are fairly big. But it's a fair point. You absolutely could have this problem. Because if you look at two different lines, maybe you recombine the cis regulatory regions. But I can also do this by taking this cross to the tester and comparing two different environments. And in this case, we did two environments. We did newly eclosed, so basically 0 to 1 hour newly eclosed, and then we did 3 to 8 day adult females gonadectomized. Okay, so the newly eclosed we don't gonadectomize because they're smushy, so it's kind of impossible to do that. But for the 3 to 8 days, we gonadectomize them. We say, okay, these are exactly the same genotypes, so even the RIL is the same, right? But we want to then compare the 0 to 1 hour versus the 3 to 8 days. So that's like a true environmental difference, right? So we can then have the trans effect due to the background genotypes being different, and then we can have the trans effect due to the environment, and we can have the cis effect due to the genotype, and all three of them we are using the exact same statistical test. Okay, so our experimental design was we had 4 recombinant inbred lines. 2 were high for thermotolerance, 2 were low for thermotolerance. We crossed them to the tester. We had the 0 to 1, the 3 to 8 day. We have females, 3 replicates of each. Okay, so that's overall 24. 128 million, more or less, long reads. So that's actually quite a number of long reads. One of the things that I told you, and I won't show you all the details, but one of the things I told you is you get a lot more information out of this. So this is a more or less equivalent to about 10 times the number of short reads. So more like 1 billion short reads. We first got the long reads, though it was, I'm just going to say it was challenging to deal with them. because there were not a lot of tools kind of set up to tell, like, well, did my experiment work, and are my reads quality, and are they long enough, and how do they look, and so on. And so I don't take a lot of time to talk about it today, but I just want to mention that a PhD student of mine, Natanya Keel, she developed a QC process for long reads to be able to enable you to kind of look at your long read RNA-seq. So if you're going to do RNA-seq, but you're going to use long reads, and you want to know, like, how do my samples look, and are they comparable, and is the quality similar? She has a tool for this. I told you that we have this pipeline Bayes ASC, and it's all there, and you can use it, and so on, and it's true. It's a bunch of Python scripts. We have packaged them up nicely in bash shells. We've made a next flow. It's very good. Like, if you're computationally sophisticated, you're going to be great. Doing that, I really learned something I'm not good at, and that's software. So writing software is like a whole thing in and of itself. It's not like creating an R package and sending it out, which is more or less what we did with Bayes ASC. We have an R package that you can use for that. So Anna Conessa, who has been a recent collaborator of mine, is a bioinformatician and really is somebody who builds software. So just to say, this is a next level kind of tool that has been built really because of Anna and her group. Okay, so now the big results. So in red, we have when we have different haplotypes. So basically the cis effects. In blue, we have when the haplotypes are the same, so the trans effects. And then each pair of bars is a comparison between the two RILs. So if I have 4 RILs and I have 2 pairs, that's 6, right? 4, 2, 2, 6 combinations. And for each pair, I can say, if I compare these two to each other, or they have the same haplotype, or they have a different haplotype, I'm going to do a test for cis, or I'm going to do a test for trans. And then I can compare what happens at the 0 to 1 hour versus the 3 to 8. I'm going to back up here a second just to show you something. And I want you to look here at the top left. Okay. At the top left, this is about the number of reads. So one of the things you can see is that in the 0 to 1 versus the 3 to 8, we have many fewer reads in the 0 to 1 versus the 3 to 8. Okay, this was one of those things, right? Like, we actually didn't plan to do 2 environments. We were actually going to do just the 0 to 1, but that was like such a pain to collect that in the end we ended up doing more to the 3 to 8, and we were more practiced at doing the 3 to 8, and so we ended up, you know, doing libraries, and then because they looked really good, we were like, okay, we'll do more coverage. It's like, it happens to everybody, right? Like, you have your experiment, and then you kind of get an idea, and then you do something else, and then you have the other experiment. So we have a difference in the coverage overall between the 0 to 1 and the 3 to 8. So in the 3 to 8, we have a lot more coverage than we have in the 0 to 1. Why am I telling you this? Well, didn't I just spend a bunch of time telling you that we wanted to have a model that would account for differences in coverage and let us detect allele imbalance, you know, when we could account for differences in coverage? And so one of the things that you should see here is when I'm comparing these pairs of allele imbalance within the 0 to 8, or between the 0 to 8 and the 0 to 1 and the 3 to 8, my kind of detection probabilities for my cis and trans effects are quite similar. I have higher detection of trans in the 3 to 8 than the 0 to 1. Can you guys see that? The light blue, which is the trans effect, that is the same haplotype being compared in the 3 to 8. This is bigger on the bottom set of bars where I have 3 to 8. So my cis effects are similar between the two, and my trans effects are more. What does that tell me? It tells me that my effect sizes for the trans are also smaller. So I need more coverage to have more power to detect them, whereas the cis effects are pretty big, and I tend to detect them even at the lower coverage levels. Okay, so what does this look like if I kind of add it up across all 6 crosses? Okay, in the blue is, so this plot has every gene in it, as opposed to here, each gene is going to be there depending on which cross. So the same gene can be in this, the same gene can be in this bar, 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 the same gene, right? Because I'm just going to compare between those 2 crosses. But I wanted to say, okay, at the gene level, does my gene have evidence for genetic variation in cis? So I can look across the set of crosses and say what's happening at the gene level. And if I look at this, okay, what I can see, 2 things. It's dark blue if I see the evidence of cis in more than 1 cross. So what this means is that most of the cis effects that I'm seeing, I'm crossing, remember, I'm crossing these recombinant inbred lines to a tester. I probably should take a moment now to talk about this a little bit more, about the tester and about why I want the recombinant inbred lines against the tester. So the recombinant inbred lines, they came from their own kind of population and they got extracted. We picked a tester that was different from all of them to try to maximize the cis differences. Remember, I told you I can only find trans if I also have cis. So what we really wanted to do was like maximize the amount of cis differences that we see. So try to pick something different from the recombinant. Correct. There's also white 118 so that we could see if we had contamination in the F1s. Just as a. So what we see is then when we see a cis effect, we see it really in multiple different combinations. So when I'm comparing it, I'm seeing it more than once. I'm seeing the fact that I have a difference in this cis regulatory region from more than one source. This middle bar here, this is the trans effect due to the environment, that 0 to 1 versus the 3 to 8. And again, the dark is when I see it in more than one case. Now, the 0 to 1 versus the 3 to 8, I would think, we thought going into this, we were like, okay, we may not see any big trans effects at all because our coverage was so different between the 0 to 1 and the 3 to 8. We thought, oh, you know, we're going to have very little power to see those differences between the 0 to 1 and the 3 to 8. But actually, we see more differences there than we see when we compare the 2 lines within the 0 to 1 or within the 3 to 8. Okay, so this is really giving us a sense that the trans effects are smaller than the cis effects. And not only are they smaller, the genetic effects of trans are smaller than the environmental effects. So if I think about comparing, for example, two tissues, I might expect to see pretty big trans effects. So here we're effectively, I mean, these are not really hugely different environments. I'm just going to say, right? It's a newly closed adult female versus a 3 to 8 day virgin gonadectomized. And if I do differential expression between these two, there's like, I don't know, 50 genes differentially expressed between them, like almost nothing. Like they really look very similar in terms of the environment. And yet we see a pretty big trans effect difference between these two. That is really saying something about how trans really drives an environmental effect. And to me, it's kind of interesting because you have in the literature all these arguments, cis, trans. And if you stop and think about it, whether or not you have expression of the gene, like I have it, I don't have it, that's a trans effect. So if you just counted all of the differential on off between two conditions as a trans effect, in this particular case, we'd have like 750. They'd be up here. So now if I added up all of my trans effects, like the trans effect of being on off, the trans effect of the environment, and the trans effect of the genetic, like if I put all those together, they're actually pretty similar to the cis effects. And so the question is, how much of these cis effects these really overlap? These trans effects, they look like they overlap a lot less. It's also true between the environment and the genetics. There's much less overlap between what you're picking up in trans signals in the environment and the genetics than there is when you see these cis effects overlapping, which I think is a really interesting message. And why I started out by showing you guys, oh, this transcription is complicated, right? Because you have the cis effect, but then you have all of these other things happening in trans, right? And so all of those pieces can contribute, but probably like the model would suggest none of them very much. All right. I'm going to take a kind of a walk on the wild side here just because I'm talking to PopGen people who understand how crosses work and say when we first started thinking about this, we started thinking about how you could also think about your environment as being different in a reciprocal cross, right? So one of the things that happens in flies, like if you do the reciprocal cross, right, is you have a different mother versus the father. So even though you inherit this chromosome and you have like, you might be looking at the exact same genotype, nuclear genotype, you're going to have inherited the one allele from mom and the other one from dad. Now, this is a study that they published all the data for in mice. So mice have imprinting, flies do not so much have imprinting, but mice have imprinting, and they did a fairly back of the envelope calculation about whether they had a parent of origin effect. And we really wanted to see like, well, can we test actually for the difference of this? Again, using that kind of comparison of the allele imbalance differences between the two. And the answer to that was yes, absolutely we can. The other thing is if I really wanted to evaluate the cis effects, right? Do I have to actually cross all the alleles pairwise? So this is a diallel design. Everybody crossed with everybody else, which they did because this is the three of the founders of the mouse collaborative cross, which is a kind of a classic design where they pulled these multi-parent recombinant inbreds off of them. So they did this kind of diallel design off of three of the parents that were the most different, and they did all the allele combinations. We wanted to ask the question, like, do you actually have to do all the allele combinations? And I really want to kind of underscore now, like, if you kind of sit here and look at it, you have, for example, if I compare these two, they share the blue. So again, I come back to having, well, this is going to be a cis test of the difference between orange and light blue. Or these two, this is going to be a cis test of the difference between light blue and dark blue. So you don't actually have to directly compare them. You can do it indirectly, okay? And then you can compare the direct versus the indirect. And one of the things then to think about is when you do it indirectly, you can actually look at progeny of the same sex based on how they are inherited also from the same sex of the parents. Okay, so let me go back over here and just say, if I want to look at these, I can actually compare because this is, let's say, mom, right? I can compare these two to each other and they have the same maternal effect, okay? And I can compare female or male progeny off of those. Okay, so this is my argument. Another message just for Christian. I think the test cross is underappreciated. If you are doing some kind of experimental evolution or you're doing some kind of thing and you have all of these different chromosomes that have come around and you then want to evaluate them and you think, oh my gosh, like it's going to be a nightmare. I have to cross everything with everything else. Well, actually, no, you don't. You can just, instead of crossing everything with everything else, just like n minus 2 uh, n minus 2 times n combinations, you can just cross everything to a tester and you can get really good estimates of what is the effects, because the tester will act as a balancer for the entire genotype. Practical purposes, we tend to use the testers as a female, just because you can make a lot of it, and otherwise it's kind of a pain logistically. All right, so now I'm kind of at the end, and I have my take-home message part 3. So allele imbalance is actually a very clever way of gaining insights into genetic variation in sort of expression. So if you think of expression as a phenotype, allele imbalance is just a super cool little trick to give you an insight into whether or not there's genetic variation that is in play. And I really want to say thank you to Patricia Whitkop and Andy Clark, who kind of came up with this idea, and 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 I have been making a career out of looking at allele imbalance almost since they came up with the idea. So it was just very nice. I can tell you that doing the allele specific expression, it can be kind of messy. Like you have a lot of different steps, different things going on. You have to map, you have to count, you have to keep track. We have some tools that work. Christian had tools for a long time. I tried to get him to update them, but did not succeed. Um, and we're happy to help. And the Bayesian component of this is actually an R package. It's in Stan, which is kind of this add-on to R that lets you do the Gibbs sampler. So that's very nicely done. And then I really want to challenge this particular group to thinking a little bit about what experimental design you really want to do, like what cross can you make. We used to do this all the time, like way back. Think about, what do I want to cross? How do I want to make that cross? How is that going to tell me what I need to know? I think you have a lot of resources here, and you can really think about like what are the culminations, the experimental design, not just in terms of like how to keep your densities uh, um, constant and so on, but really like what kind of genetic material you want to produce can really help you get some interesting insights. And I would say I would encourage you to think a little bit about the test cross, because it can be quite efficient at helping you identify the allele variation. Okay, so now I'm finished my formal presentation. So again, I want to thank the team